hope you can see. So I'm a pediatric oncologist. I work at the Alberta Children's Hospital. I was trained, got my medical degree from McMaster and trained at Sick Kids in Princess Margaret in Toronto. So you guys probably know overall children with cancer do so much better than adults, 85% cure rate. Most of the kids get better, go home, come back and see us during Christmas. But there are still about 15% of the children we don't cure because we don't have a treatment for their cancers. These affect mostly the children, very small babies with cancer, and older teenagers are very hard to cure. This is about 400 kids in Canada alone every year being told, you know, we have nothing more to give you, just go home and take care of yourself.
I would ask everyone else to.
Yeah, I saw you there. I saw you there a minute ago.
Okay, so a few years ago, I had to give this talk to a parent that the little baby had an incurable brain tumor that didn't respond to any of the treatments that we had. But the mom asked me a question I have never been asked before. She said, I know there's going to be a cure for this cancer. Can you please keep my child alive until that becomes available? So essentially at that point in time, just started this project. And I went home and I told my wife that I'm switching careers and we are going to be poor. So anyway, so what we did going forward was we managed to get some of the tissue from international collaborations. We worked on the biology. We identified exactly what drive these tumors, the pathway. We published the very first time the mechanism behind these tumors in Neuro-Oncology Advances. Then we went on to look at how we can overcome the effect of these tumor-causing proteins in these tumors, in these cancer cells. So we went to the computer, designed molecules that can bind effectively and neutralize the function. We tested those drugs over many, many years. And finally, we found an agent that was very effective in targeting this protein, causing tumor regression, tumor cell death, without causing any interaction with normal cells, because it is so unique and so specific. This is an example of what we have done with some of the animal models. We treated mice carrying these tumors with our drug and with placebo. As you can see, that it causes a tumor regression without causing any side effects on the animals, leading to longer life. So what we have done subsequently is we looked at large databases and found exactly the same pathway is also active in many difficult to cure adult cancers, including breast cancer. And as far as we know, there are no other drugs in development or already being actively used in the clinic to target this pathway. So our team, we have postdoctoral fellows and technicians. We have the educational background experience in developing new therapies and some commercialization expertise, as well as the skills to manage such a project going forward. So this is where we are. We are much earlier stages than many of the presentations we heard before. So we have completed the discovery, which includes early pharmacology and on-target and off-target effects. And importantly, we have also found the mechanism of action of our drug exactly. And we have completed the preliminary safety studies in animals. And we are in the process of finding a way to synthesize this drug in a GMP facility. Right now, it's made in a chemical company in Edmonton, where we are looking to synthesize this molecule in a GMP facility going forward. And I've already talked to our adult oncology colleagues at the Tom Baker Cancer Center about including some adult patients in this clinical trials. This is because right now the treatment for these adult cancers are very expensive and very, very toxic, including bone marrow transplantation costing hundreds and thousands of dollars for treatment. And so going forward, we would like to complete the initial critical clinical trials and then look into acquisition and licensing by large pharmaceutical companies that can take this very important drug forward to clinical trials across North America and probably across the globe. So what we ask is an investment to complete the GMP studies as well as the early phase clinical trials, and support to navigate this process through and advice on negotiations with large pharmaceutical companies going forward. So thank you all very much, and I'll be happy to answer any questions that you may have.
I guess I can start off the round of questions here. So one issue that's pretty prevalent among cancer treatment is one, drug delivery, and two, specificity. How does the drug you have developed combat those issues?
Yeah, so a very important question. So the very first one is the delivery systems. So in our animal models, both spontaneous as well as xenograft models, there we have shown that this drug is orally bioavailable, and also importantly crosses the blood-brain barrier, so it can be used to treat brain tumors. We have done the pharmacodynamic and the pharmacokinetic studies in these animals. Sorry, what was your second question?
It was about the specificity of the drug to tumors.
Absolutely. So we know this drug is extremely specific to this altered molecule. So mechanistically, what happens is that when we are growing it in the embryo, we should allow cells to proliferate and grow. That's how babies are made. But that process should stop when we are born, but it comes back when a tumor forms. So in 2 times in your lifetime, you have excessive proliferation, is one as an embryo, the other one when a tumor forms. So during embryonic stage, this protein is suppressed and to allow cell growth, but somehow it doesn't go away. What our drug does is binds to this protein and makes it inactive, so the tumor suppressors in the system will function to prevent tumor growth. So we know that the structure of the chemical binds, as far as we know, through molecular analysis only to this particular protein because of the binding specificity in the groove of the protein. And we have done the non-specific studies on normal bone marrow, normal white cells, and normal brain cells, and so on, shown there's no toxic effect on those cells.
Very impressive talk. If there's other questions, I'll ask a quick one. I'm just wondering about the possibility of getting grant funding, CHR, other funds, rather than investment. What's your strategy? What's the ideal balance there? I know it's not easy in Canada, but is there any possibilities to get some grant funding in to help bridge this early stage?
Right. We have applied and got the patent for this drug, and we know that this drug does not exist out there. It's a very unique structure. But the feedback I got is that when you write a CIHR, I do have a CIHR for related but a different project, that you have to disclose some of those information. And again, I'm looking for advice on how to navigate that process right now. So that is kind of a tricky line to walk, how much disclose, because they have to know what they are funding. That's fair. But on the other hand, how much we can disclose.
Sure. Yeah, I think that there's a number of companies in the Alberta space that are in that dealing with that. So you might get some feedback from maybe Len and others. I think it's a frequent problem, but are you designing more molecules based on structures as well? Do you have series that you're exploring, or have you really fixed your lead candidate at this stage?
Right. I mean, this lead candidate came out of a screen of about 100 molecules that we had to make. Some of them were very active, but not soluble, not crossing the blood-brain barrier. After screening, one thing that we have for our advantage is that we made some cell lines from these extremely rare tumors that allowed us to screen and select these molecules. So right now we are focusing on this molecule in the process of doing some large animal studies to confirm the non-toxic nature of this molecule. Right now we are focusing on this particular compound.
Okay, thanks. There's a couple of great questions in the chat space. John Porter, maybe I'll ask for you, is this across all cancer types? And your pH 1 N equals 10, is that enough for stats? Just take a look at those questions. Maybe you can address those.
Yeah, so yes, this pathway seems to be tumor agnostic for some reason. We have an idea, but it seems to occur more in the cancers that occur in newborn babies or during the early childhood years. Interestingly, it also comes back when somebody is 95 years old and develop a cancer. We don't understand the mechanism behind it, but it does happen across all tumors. In a smaller percentage in breast cancer, we found about 5% of the breast cancers, but almost all of them are non-responsive to current treatment. It still translates to a big number. So right now, if we have the target, then it's active and it's tumor agnostic.
Okay, John, if you want to ask any further questions, or Tanvir or Jeremy, we have one more minute if anybody wants to pipe in.
Yeah, I'll pipe in. Just a follow up to that. You're doing your N is 10 patients in the phase 1. So I'm curious if you can gather any PK/PD data for that moving forward into phase 2.
Right. So this is after conversation with some of my adult oncology colleagues. I do phase 1 for children. That's my major job, and it has to be multi-center. And these are extremely rare tumors, so very hard to accrue a number of patients to make any biological data out of it. But in adults, this particular tumor type, relapse and refractoriness is very, very common. So I was told that N of 10 will give us information on the toxicity by definition, because it's a phase 1, that's no efficacy assessment, but you can gather some data and definitely do some PK/PD, but that would help us to put together a much larger multi-center clinical trial to acquire the appropriate statistical information. Again, that'll be a point where I may be able to talk to large pharmaceuticals, because it's extremely expensive to do multi-center clinical trials, especially in pediatrics.